It modulates activity of PI3K-AKT signaling pathway. Dihydroartemisinin, this is one of the very important molecule derivative of artemisinin. This mainly targets p53 and JAK-STAT dependent pathway. Tolite, it also works p53 and FKB and sirtuins. Ginseng is one of the finest plant molecule which is available in traditional Chinese medicine and Indian traditional medicine, which is utilized for its values as antioxidant and inflammatory molecule, especially because this especially targets p53 and FKB, PI3K, AKT, ERK signal. Another important component is curcumin that comes from curcuma, that is one of the very rich plant in India, that is yellow colored substance, and it has been widely utilized for modulating Wnt beta catenin signaling pathway as well as p53 dependent signaling pathways. So, natural plants have a diversity of their mechanisms, and as we have discussed, the three driver genes, ERK, p53, EGFR, and KRAS, they are going to work on all the signaling pathways. So, this is a summarized list for some of the compounds like green tea, isothiocyanates, gemstane, pomegranate, quercetin, curcumin, and indole-3-carbinols. They mainly target p53 dependent signaling pathways. So here we have the scope that which compound can be utilized along with one of the therapeutic agents for targeted drug delivery as well as combinational therapy. This is another complicated picture in which there are some of the compounds like zerumbone, tomatine, sulforaphane, PFE and quercetin, tomatine, curcumin, lyseolin. They all are working at different levels of cell signaling pathways. And most importantly, they have p53 dependent activity. Therefore, ultimately, they can inhibit metastasis, they can induce cell cycle arrest, they can suppress cell growth and proliferation, and they can inhibit mutation. And that is what is our major aim, to design and develop some of those medicines from natural resources who can target cancer cells in a very particular manner. And this is one of the finest way to do that. This is another picture in which some of these compounds are shown to directly interfere with p53 dependent pathways, which mainly target AMPK, PI3K, AKT, and p53 dependent apoptosis, gene instability, cardiotoxicity, etc. So these molecules have ample scope to be utilized in drug development. And there is a long list. I'm not reading all the list. It can be shared for discussion. That there is a long list of compounds from natural resources who can be very suitably applied. This is slide 1 and slide 2. This has a very long list of compounds. Now, the second pathway, that is EGFR, it is very interesting. The full name is epidermal growth factor receptor. This belongs to ERBB family of receptors of tyrosine kinases, and they are onto the cell surface, and they have different cellular sensing and signaling mechanisms. They mainly act by phosphorylation activation, and they have their adaptor proteins, they affect some enzymes, they modulate the transcription factors, and ultimately they regulate cell growth, migration, invasion, etc. And once there are mutations in EGFR that we have just discussed with that, it causes aberrant activation of tumor. This is the detailed pathway where EGFR targeted signaling is utilized. And these are some of the tyrosine kinase inhibitors like afatinib, dacomitinib, erlotinib, gefitinib, and osimertinib, which are targeted molecules working in this direction. Now, more interesting is that some of the natural products They also have very similar activity like tyrosine kinase inhibitors. They modulate autophagy in EGFR dependent signaling, VEGF expression, apoptosis related EMT, cytokines, chemokines, etc. And one of their target is EGFR. And there are a bunch of other bioactive compounds who can target EGFR dependent signaling. They are belonging from alkaloids like tetranidine, matrine, homaragine, polyphenols like resveratrol, gallic acid, dorsifol, ellagic acid, and one of them is epigallocatechin 3-gallate EGCG that comes from the green tea. These polyphenols are one of the fantastic drug family which can be utilized in trials for their effect against EGFR dependent signaling in lung cancer. There are different types of saponins, and one of them is ginsenoside, and the terpenoids like cucurbitacins, ornithine, artemisinin, tripalidine, lupiol, etc. So we have a variety of drug compounds which can be utilized in lung cancer treatment. And this is another extended list. The very interesting part is that these EGFR targeting natural products, they can be utilized for chemoprevention through EGFR dependent signaling as well as in chemotherapeutics. So they have both the ability. So from these basic researches, they can be utilized for extended application in this particular therapeutic domain. So these two slides, they are from those natural products who can target EGFR dependent signaling in lung cancers. The third signaling pathway of the driver gene, that is KRAS, that is basically Kirsten-Rack sarcoma viral oncogene homolog. This is one of the group of genes involved in the pathway of EGFR dependent signaling. So EGFR and KRAS, they work almost together, okay? And they modulate three major things. One of them is NFKB, nuclear factor kappa B, ERK, and mTOR. mTOR regulates autophagy, ERK regulates cell signaling, and NFKB, so ERK regulates cell survival, and NFKB regulates cell proliferation and inflammation. So collectively, they regulate cell growth, proliferation, and survival. They interact with several signaling events in the cell, and the tumor cell can be invaded if there are a mutation in KRAS and EGFR, and that's what happens in the lung. When there is an exacerbated form of lung cancer, that is non-small cell lung carcinoma, that is around 85% of the total type of lung cancers, while the small cell lung cancer is only 15%. They have extensive mutations in KRAS and EGFR. So these are the list of the downstream target genes who are dependent on the KRAS dependent signaling. PI3K dependent, PI3K AKT dependent cell survival, RAF, MEK, ERK dependent cell proliferation, RAL, GFP, NFKB dependent cytokine production or inflammation. So if you alter the expression of either of the genes or induce mutation, that happens in case of lung cancer. So it will cause aberrant cell proliferation, aberrant cytokine production, and inflammation. Therefore, tumor formation, tumor transformation, epithelial mesenchymal transition, and invasion metastasis. So there are a variety of natural products that are utilized for KRAS-dependent signaling pathways. Many of them are very particular to inhibit RAS processing. For example, these are some of the names. These are mainly belonging to artemisinin, terpenoids, and polyphenols. And some of them are very specific to act upon RAS-dependent activities which is associated with proliferation. And some of them is RAS-dependent activity associated with survival. Collectively, they make a group of those compounds which make it a very critical component in terms of suppressing cell proliferation and metastasis. This is a long list of KRAS targeting natural products in lung cancers, which can be utilized for regulating lung carcinoma. So this is the list, and these are the effector genes which are basically modulating apoptosis. So with this, I can conclude that these are some of those compounds which can go to clinical trials and research in clinics for utilization against lung carcinoma. And this is just a cumulative summary that it's one of the treated disease which is an NSCLC accounts for around 85% of the cancers. And it can be treated with the help of combination of anti-cancer drugs along with natural products like grapeseed extract, proanthocyanins, green tea extract and EGCG, curcumin, parpinolides, resveratrol, silymarin, ginsenoside. They are from different type of traditional Chinese medicines and Indian traditional medicine. So with this, I'm thanking to the organizations for giving me this opportunity to speak, and I'm open for the discussions. Thank you so much.